Going to take a second and start the recording. All right. For everyone that's online, I just want to let you know that your mics, cameras, and screen shares have been turned off. We ask that you keep them that way during the duration of this event, but we do hope that you ask questions and that you do that through the chat function on Zoom. Please do that anytime you have a question. We'll work through them as they come in or at the end, depending on how the questions kind of play out. I wanted to let you all know that we are recording this seminar, and I'm going to put the link on where that seminar will be posted in a little while, probably about 2 days. It takes me to process that. So if you're interested in watching this again or sharing this particular presentation out, you can do that using the link that I just put into the chat box. Also wanted to just let everybody know that we will not have a formal seminar next week, but we will be celebrating our graduate students at our annual Markham Symposium. And so the Markham Symposium is a place where our graduate students get to share the work that they're doing and talk about upcoming work. So if you're interested in participating with us, we encourage you to do that. It is being hosted by HUVA. And I'm hoping maybe Yichang can put a link to that into the chat box as well for folks. If you're interested in participating, you can go on to that link and learn a little bit more about the Markham Symposium that will happen next Thursday instead of seminar. So if you're interested in that, please join us. If you are interested in any of our Hatfield events, please go to HMSC's homepage and go to the bottom. There is a calendar of events there with everything you need to know about upcoming events and how you can log into them. I also wanted to, I do this every week, but I'm going to make another plug. We're still booking some seminars. So if you have anybody that you would like to hear from, that we'd like to invite to speak with us, that would be great. Let me know and we will invite them. We've got some openings for the into September. So please let me know if you've got somebody you would like to hear from. All that being said, I just see that Yichang put the link to the Markham Symposium in the chat box. Thank you, Yichang. So if you're interested in coming to next week to the Markham, that link is there on how you can register. For today, let me tell you a little bit about our speaker. Dylan Gomes received his bachelor's in biology from the University of Montana and holds a PhD in ecology, evolution, biology, and behavior from Boise State University. He is currently a postdoctoral research with CIMRS and NOAA's Northwest Fisheries Science Center here in Newport. We haven't seen him around because we haven't been there, but we're excited to have him here in Newport with us. His research is focusing on building an ecosystem model of the Northern California current ecosystem with a special focus on understanding juvenile salmon survival in response to ecosystem perturbations such as climate change. So welcome, Dylan, to HMSC and to today's seminar. The floor is yours. I'm going to have you take it away.
Thanks, Simon. You can hear me?
Yes, you sound great.
Great, thanks. So yeah, thanks everyone for coming. And so as urbanization continues to spread, a lot of things come with it. And a couple of those things are human-made lights and human-made noise, or what we call anthropogenic noise. And lots of animals, from cuttlefish to birds and whales, bats and insects and spiders, all respond to anthropogenic noise in negative ways. Most of this work has focused on birds and mammals, particularly bats and cetaceans, with some pinniped work. And so birds have sort of become a bit of a model organism for noise research. But the point here is it's been a little bit biased towards some groups and not others. And it's not really a surprise that animals are affected by noise. Right? Animals use sounds for all kinds of things. So they use it for communication, they use it for foraging, avoiding predators, and in daily life. And acoustic communication has also sort of received a lot more attention than some of these other things, but it affects more than just that, including survival and development and others. And a lot of animals have responses that appear to allow them to mitigate or deal with anthropogenic noise. Right? So everything from insects to frogs to whales can increase call duration. They can change the pitch or the frequency of their calls. They can increase the amplitude of their calls. and all sorts of strategies to deal with anthropogenic noise. And that's, unlike or not unlike humans, where if we're trying to talk in a really loud, busy place, we focus a lot more on lip movement and body gestures and things like that. So we use alternative sensory modalities, and we talk louder. It's called the Lombard effect. And so there are a lot of strategies that we all use to try to communicate in really noisy situations. And so as urbanization expands, this is more and more of a pervasive issue. And humans tend to not think about this so much because a lot of us were raised in and live a lot of our lives out in these really noisy urban environments. So it's not at the forefront of our mind all the time, and we're pretty visual creatures. So it's something that's been relatively ignored until recently. And it might be part of the reason that a lot of people are starting to seek outdoor quiet spaces, natural soundscapes, is what they're called, to get away from sort of that noisy din. And there's a lot of evidence suggesting that humans derive all sorts of psychological and learning and just mental state benefits from being outside in natural soundscapes. But not all natural places are really quiet, and some of them are very loud. So this is a photo I took north of Boise, Idaho, which is where I did my PhD. And this is the North Fork, the Payette River, and it's incredibly loud for most of the year. And unsurprisingly, animals also seem to be able to deal with natural noise, natural meaning non-human made noise. So birds can deal with crashing surf, penguins can deal with wind noise, and frogs can deal with the chorus of other frogs. And understanding, if we can understand how animals are dealing with natural sources of noise, right, that they've spent much deeper evolutionary history sort of evolving to cope with, then we might be able to understand a little bit more about how animals are dealing with anthropogenic or human-made noise today, right? Because this is a really small sliver of the entire sort of timeline of how animals have been dealing with noise. Hearing organs evolved a long time before traffic noise, for example. And so we sort of have this dichotomy, or this, it's sort of a false dichotomy, but this dichotomy between natural noise and anthropogenic noise. How are they different? How are they the same? And so to sort of get at that, we're going to kind of go on a thought experiment here. So if you're standing below this dam, this is a dam below Flathead Lake in Montana. Is this source of noise natural or anthropogenic or human? Well, the sounds of rushing water is what we think of as a natural source of noise, but humans happen to have put this dam here in this specific geographic location and have changed the soundscape. Right? So maybe the lines are starting to blur. And what about human-induced climate change, where the amount of snowpack and the snowmelt rates and winter storms and the entire timing of stream runoff changes? Are the changes to those soundscapes, are they still natural or are they anthropogenic? So we sort of need a new way to think about it. And I would argue that the new way to think about this is in a quantitative way, right? We can measure different aspects of sound. So we can measure frequency, which is the number of pressure wave oscillations in a second. So we perceive this as pitch. So pitch and frequency are the same thing. We could also look at amplitude, the intensity of that signal, which is perceived as loudness. And then we can look at the temporal patterning of either of those two things. And so to look at some examples, here's a spectrogram. This is of a river. This is indicated by the three blue lines at the top. And so time is on the x-axis, the frequency is on the y-axis. And the dashed white lines here are the range of human hearing. So anything below the sort of central band there is infrasonic, and everything above that is ultrasonic. So you can see that there's a lot of noise in the infrasonic range, but it extends up into the ultrasonic. And so this recording was actually taken right by the photo I showed in the beginning. And so then we can look at what's called a power spectrum here. That's sort of it's typically viewed the other way, but it's tilted on its axis here to align the y-axis. And so this is looking at the same information, but we're sort of looking at, instead of having a color-coded amplitude profile, which is what the spectrogram has, we're turning that on its side and looking at the amplitude profile sideways. And so this is what a road looks like. It's one recording of a road. Roads can look different depending on the types of vehicles and how much, and the texture of the road and all sorts of things. But you can see here that roads are a bit more intermittent, right? They're not as continuous as this river, at least. And so the reason this is important really comes back to acoustic communication. So this is another power spectrum. It's just sort of like a heuristic here, but it's flipped back on its axis. And so frequency is on the X. So, higher pitch you might think of as we get towards the right side of that axis, and the y-axis is sound pressure level or amplitude. Think of that as intensity. And so the little blue polygon here in the middle is, a vocalization of a human or a bird or a frog, and the gray is the background noise. And there's something called a critical ratio, which we'll just distill more simply into that is the amount that your signal has to stick out of that noise before you can hear that signal, right? And so in this case, this signal, this blue signal, if I was the receiver and this sender sent this signal, I would not be able to hear it in this particular background noise profile. And so there are lots of ways to solve this. I talked a bit earlier about how animals solve this noise problem. So if we look at the left column, you could simply signal in a less noisy area. So you notice that the only thing that's changed there is that the gray background noise has dropped a bit. The middle column, you can signal more loudly. So in the top of that middle column, right, the blue signal is quieter. It's got less sound pressure, lower sound pressure level, and that has been increased in the bottom plot. And then at the right, you can signal at a higher frequency. So that's been shifted up to the right. And so what's really cool about this is then, with experimental biology, you can filter noise in different ways to both what's called mask or not mask a signal of interest. And so here, these gray shaded boxes are showing background noise profile that is either not masking this frog call or masking this frog call. And so what we did for my PhD project is we decided to take this idea and apply it across the landscape, with the idea that if we play low frequency noise, so here on the sort of left heuristic, that it might filter out organisms that use lower frequencies more often. So you notice that the crow and the woodpecker and the robin aren't in this lower left section, whereas they are on the right. And so in this right panel, the black here is indicating noise that's been shifted upwards in frequency, or noise at a higher frequency. And so the idea here is perhaps if vocalizations are important, right? Because we know noise is changing things, but we want to know why. And that can be a lot of different things. And the prevailing hypothesis is that noise is important for acoustic communication. And so this is a direct way to test acoustic communication across a landscape by having this hypothesis that if the background frequency is overlapping with the vocalization, the song, the call of that animal, it should filter out that animal. And so to just show a few of the spectrograms from the field work, and then I'll get into the methods. It kind of seems a little backward, but just trust me, it makes sense to present it this way. We have three spectrograms here, and so you can see that the frequency is increasing as you move to the right, but the amplitude is also increasing, so the color is getting brighter. Right? And so this image here on the bottom left is a quiet stream that might be, it might look something like the spectrogram on the left. And the bottom right river, as I showed you before, is going to be really loud, and so that might look like something that's on the right. And so what we did experimentally is we hiked a bunch of speakers out to these sites, and we were able to turn them on. Right? And so this top right little spectrogram insert here shows that when the speakers are off, the river is really quiet, and when the speakers are on, the river appears to be pretty loud. And so essentially what we did for this work is we turned a small stream like this one on the left into one that sounds like this one on the right, even though all the other environmental variables were kept constant. Right? So sort of the power of the experiment is you can just tease apart the role that the soundscape plays in shaping animal communities, distributions, behavior, without all those other sort of confounding variables like more water moving through an area. But the other advantage of the experiment is then you can also shift what frequencies you're broadcasting with those speakers. And so this is a whitewater river noise spectrogram that's been shifted upwards in frequency. Right? Again, we want to understand if we are filtering out animals based on their vocalization frequencies. This is sort of the experimental setup here. And where we went is, or where we brought these speakers is out near Craters of the Moon National Monument. It's a couple hours east of Boise in Idaho in the Pioneer Mountains. And so we selected 20 sites, and at some of these sites we played back noise. So that's the squares here are where we played back river noise, and then the triangles are where we played back that shifted noise. And the circles are all sort of a range of controls. So you can see some of the circles are really, really red, which means they're really loud. Some of them are pretty white, which means they're pretty quiet. And so all of these sites had rivers to begin with, but some of them have been experimentally altered, just the soundscape component. And so to do this, it required a lot of gear. Right? So we had multiple U-Hauls, multiple pickup trucks, a lot of batteries, a lot of this electrical conduit that we suspended speakers from, a lot of solar systems. So these are all solar controllers. You can see some stack of solar panels on the left here and a bunch of wind screens in a pile just to the right of those. And so just a lot of gear. So all these batteries that were able to power our sort of off-grid speaker systems. And in total, it was about 4 tons of gear. And we hiked all of this in, or I should say most of this in, on our backs. And so a lot of these sites are a couple miles from a road. Some of them are close, but most of them are a little bit further away, which is pretty intense. There's it's a lot of bushwhacking, a lot of weight. But we did have a road that got washed out. It would have put some of our hiking quite a few more miles. And so we had the opportunity to rent a mule pack train of mules and a mule packer. And so you can see myself and a postdoc in our lab sort of strapping some solar panels down to this mule. And this is sort of what the landscape looks like a bit here. So sagebrush with these little riparian draws of cottonwood and aspen, willows. And this is what a speaker looks like. So we have the solar panel here in the foreground and the speaker up on a tripod in the background. And the solar panels allowed us to charge the batteries, which allowed us to play this noise 24/7, which is really important if you are interested in how these communities might be changing, because in nature, river noise just doesn't turn off overnight. And it also allowed us to play noise during the entire breeding season, or for 3 months. And so part of the important thing was getting out there before bats got out of migration and hibernation, and before birds migrated back in. We wanted to get the noise on the landscape to sort of, once birds make a territory decision, it's a lot harder to get them to move. And so we wanted to sort of see how animals were selecting habitat based on the soundscape. So we had to get out there early. And so this is a bit of a heuristic of what the experiment looks like. So each site had an array of 3 speakers along some small moving stream. And then we point counted birds at 3 locations. So one of those was sort of right in the center of those sites where it's the loudest. And then one of those was, a really far distance, and then one intermediate distance to try to capture a range of sound pressure levels and frequencies. And we also paired all of these with bat detectors. So each of these locations had their own little tripods with ultrasonic microphones and loggers. So here's a picture of one of those as the sun's setting. And so here's a bit of what the difference in speakers on versus speakers off. So on the left, we just have some density plots of the sound pressure level. Right? So 60, 70, 80 is incredibly loud. 30 and 40 is pretty quiet for us. And so the colors here are different sites, or are different treatments rather. And you can see that before the speakers are on, our sites appear to be pretty randomly selected in terms of, there's a lot of overlap in sound pressure level. And then once the speakers are kicked on, you can see the yellow and blue density plot there on the right have been made a lot more intense. And so a last point here is that, although we're calling this a treatment here, and it's, sort of these colors are categorical, you can see that that there is a range for any given treatment. There's a big range. And so it didn't really make sense to treat these as a treatment statistically. And so the other thing that we had to do is we put all of these acoustic recording units out into the field. And so we call these things Russian hats, but they're these fuzzy little windscreens, fake fur that are protecting a recorder in there. And then there's a battery attached through a cable. And that allowed us to actually record and quantify all of these locations that we were counting birds in and recording bats, as opposed to just using this categorical, playback on or off. And so we saw about 73 species of birds. Well, we saw 73 species of birds, not all of them as common as others. And we had 7,000 observations and over 1,900 visits to individual locations. And so these are all 73 bird species here on this one slide. So quite a diversity. And overall, birds in general avoid high sound pressure levels. So the louder a place gets, the fewer birds are there. This isn't incredibly surprising, as it's just more difficult to hear as sound gets louder. And when you look at what we're calling spectral overlap, which is, it's just the distance between that individual species' vocalization frequency and the frequency of the background noise, you see that there is, as spectral overlap decreases. So notice here that the x-axis is flipped. So as we move towards the right, there's actually more spectral overlap. So 0 is perfect spectral overlap, right? And so as we increase spectral overlap, there are fewer birds as well. So earlier I was kind of talking about how, why does this matter and getting at mechanism. Well, if we go from the plot on the left, which is, well, birds are avoiding loud areas, and the question is why, and the plot on the right suggests that vocal communication might be important.
So Dylan, we're getting a couple of questions. So I'm going to jump in.
Sure.
So can you go back over and just clarify the difference between a phantom river and a shifted river?
Yeah, so you said what did you say, the first river?
A phantom river,
yeah,
shifted river?
So thanks for that question. So a phantom river is just the word, the phrase we use for the river playback. So it's confusing because they're all already streams there, but we're basically creating what we call a phantom whitewater river. And so those are just the regular, where we recorded a whitewater river, and then we go to a slow moving stream site, and we're broadcasting whitewater river noise. Thank you for that. I should have clarified. In the shifted river is it's that same recording, but we've spectrally altered it. So we've shifted it upwards in frequency to basically just assess the level of masking or the role of acoustic communication.
Thank you for that. And we're getting a couple more, so I'm just going to go ahead and ask them why I have you here. Sure. Is there a reason why the speakers had to be suspended off of the ground?
Yeah, that's a really good question, and it's about propagation. So, well, propagation and weather. So one, the speakers had to be pointed downward because snow and rain will ruin a speaker, basically, the drum of it. And so the speakers were pointed downwards, and you lose a lot of propagation. The sound is absorbed by the ground a lot. And so the higher up you can get it, the broader you can sort of area you can influence. A good question.
Okay, and then one last one, and then we'll let you go on.
Sure.
Doesn't seem like there was a great loss in species numbers. Can you please clarify? And I think it has to do with the slide you have up now.
Yeah, so I just note that these aren't species numbers here. So these are abundance of birds, and so yeah, there isn't a great loss, but it is about an 8%, 7% decline every 12 decibels, and so that is, I mean, it is considerable over the sort of range we're talking about, and so it's, the other important point here is that these lines through these data are just one realization of a model prediction. So these aren't best fit lines. The raw data here are the raw data, and then there's a model fit with a lot of different explanatory variables, not just soundscape variables like frequency and sound pressure level, but also elevation and riparian vegetation and things like that. And so these lines represent if you fix all of the other environmental variables at their median value, and then predict the abundance of birds along the sound pressure level gradient that we're representing here, that's what that slope is there. And so depending on the environmental variables of a particular site, that line might move around a little bit, if that makes sense.
Thank you very much, Dylan. And for everybody that's putting in questions, keep them coming. Thank you very much. All right, back to it, Dylan. Thanks.
Thanks. Yeah. And thanks for asking questions like that. Cinnamon and I were trying to deal with some technical difficulties in the beginning where I could see my notes and I've lost them all. So I'm just winging it. So I appreciate the questions. Okay, and so this plot here is a little more complicated. This is the interaction between the last two things we were looking at. And so if you start in the upper right, you see that the title is SPL equals 80 dB. So that's a really loud area. Sound pressure level is 80 decibels. And then it goes down by 10 decibels all the way to 30. And so this the slope here, this is the same, basically the same plot as the one on the right from this previous slide. And so as you move towards 0 on the x-axis, from not overlapping to overlapping, the frequency overlap matters a lot when it's quiet. And so what this seems to suggest is that localization matters, but it's making a much bigger difference when the background noise is not really loud. So that's pretty counterintuitive and probably suggests that when it is really loud, there are other things that matter more. Right? There are other things that are sort of swamping this pattern, like foraging or something like that. And so another thing that we were really curious about was looking at foraging. So we glued over 700 of these little clay caterpillars to willows throughout our study area. And so at the top of this is designed to be a geometry caterpillar. You can see at the top of this little inchworm, there's a bird beak there, and it's pretty obvious to tell sort of what attacked these caterpillars. Sometimes it was birds, sometimes it was other things. And when we look at bird attacks on clay caterpillars, there's a pretty big decline over the range of sound pressure levels. And this model here has incorporated the decline in abundance that we saw on the previous slides. So even accounting for the fact that there are fewer birds in these areas, there's also a lot less foraging activity. And so for the bats, there are a lot fewer species. So we had 12 species that we detected, and we collected 100,000 bat passes that we were able to positively identify to species. And bats showed really similar declines. So it's about a 7 or 8% decline every 12 decibels. It's about the same slope as for birds. And they saw a little bit steeper decline if you look at spectral frequency. So this plot's a little different than the bird panel B because it's not spectral overlap. So bats vocalize at really high ultrasonic frequencies that aren't really overlapping with the river noise very much. And so what might be driving a lot of these patterns might have to do a lot more with foraging. And so there are a lot of animals. So there's a bat, an owl, and a fox that all use the sounds of footsteps or prey-generated sounds like the frog on the left. And what they do is what's called passive listening. So they're just using the arrival time difference between the two ears to locate where a sound is coming from. Right? And so these critters here on the right are able to do this even through snow. And so this is a pretty important use for sound is listening to the footsteps of your prey. And so noise obviously can impair that. And a lot of the bats at our study sites actually do that, and they echolocate. Right? And so echolocation is this really high frequency or ultrasonic sound that bats basically scream into the environment, and some of that sound bounces off of objects, and that allows them to tell apart a tree from a moth and just open air, right, depending on how that echo returns. And so that's a use of sound for foraging, but it's a very different use of sound than this passive listening on the bottom. Right? The frequencies are different. The passive listening is much lower frequency, typically not ultrasonic, but it can be as well. And so this gives us, another more sort of experimental power to try to tease apart what's going on with bat foraging. And so the red here is just a heuristic of some low frequency noise. And you can see that in this top panel, it's not overlapping with ultrasonic bat calls. And in the bottom, it is overlapping with whatever prey generating sound you might be interested in. And so we were able to build these foraging tests to our sites. And so here is, you can see this little red zoomed in version of this little insect wing here. We call these robotic insects or robomoths or something like that. And so this motor basically spins at a relevant wing beat frequency that the bats would be looking for. And then over on the left, we have an ultrasonic microphone. So you can adjust microphone settings to basically detect bats within a certain distance. And so there's a couple of assumptions here, but you can basically detect who is coming in to attack this robotic insect wing. And then the other side of this was using a speaker. So this little black box in the middle here is a speaker that we played cricket mating sounds and beetle walking sounds and different things from to assess how many bats or the activity of bats foraging for via passive listening. And so when we look at those, it's kind of complicated, so I'm going to take a second to walk through this. It's a similar plot as far as Y and X axes, but now we have an interaction here. And so the two uppermost for each color, the two uppermost lines are higher frequency, or they're higher frequency, but they're not ultrasonic frequencies, if that makes sense. And the blue on the left are the passive listening trials, and the red on the right are the active listening or the echolocation trials. And so in general, as the background soundscape gets louder or more intense, it appears that fewer bats are foraging via passive listening and more are foraging via active echolocation. Now we didn't track bats. We don't know if individuals were actually switching or if the individuals that prefer to forage via one modality or the other are sort of leaving and other ones are coming in to take their place. But there does appear to be some sort of switching behavior here. And so that's sort of a broad overview of the birds and the bats. There are a lot more nuanced patterns by species and things like that we don't have time to go into. But this isn't the whole system, right? Birds and bats are both insectivorous. And so we wanted to understand how their prey might be affected. And so we put six different types. We use 6 different types of trapping methods to catch insects. And so that way you can sort of catch the diversity that's out there rather than just a few groups. And we sampled insects at each of these 3 locations. But we quickly realized when we started identifying these things that we just had collected a massive number of insect specimens. So in the end, we decided to only identify these insects from the middle, because that's where we actually played the experiment. And we in total identified over 150,000 insects to the order level, which is not super refined, but it's just what you have to do with the time and resources you get. And so there are a couple of hypotheses here. And one might be that noise directly negatively affects insects that are using sound to communicate or for something else. Right, but the other is that noise is negatively affecting their predators, which then has an indirect positive effect on insects. And so we might expect to see positive effects or negative effects, depending on the context. And so these are for each order of insect and each trap type, because different trap types can catch different types within one order. So for example, Hymenoptera are bees, ants, and wasps. Pitfall traps, which are buried in the ground and catch things that just are wandering on the ground and fall into them, are going to collect a lot more ants and aren't likely to catch wasps and bees. Whereas we used some flower traps to catch bees that might be visiting flowers. Right? And so they're separated out that way. And so what we're looking at here on the y-axis is standardized model estimates for sound pressure level. So any estimate above that dashed line, that horizontal dashed line, means there was a positive effect of sound pressure level on that group, and anything below that is a negative effect. And then the thin lines are 90% credible intervals. So this was a Bayesian generalized linear mixed effect model. And I didn't mention earlier that those other ones were also generalized linear mixed effects models. And so the thin line is the 90% credible interval, and the thick line is the 80%. And so you can see that some groups were affected positively and some were affected negatively. And when we look at background frequency, you can see that a lot of them were affected negatively and some of them positively. And so you can start to maybe put some pieces together as to why they might be affected. But at this point, this is all very exploratory and very general, right? We don't really know what's going on. But it's the first assessment of this sort of thing on a landscape scale. And if you start to look at some individual groups, you see some more interesting patterns. So a similar plot, the number of Plecoptera here on the y-axis and the sound pressure level on the x, with an interaction with frequency. So here the yellow is a higher frequency and the green is a lower frequency. So as frequencies get higher, right, as they increase, sound pressure level has a negative effect, whereas low frequencies, they have a positive effect, right? It's kind of odd. And we see the same thing for trichopterans, which are caddisflies. So what's kind of interesting about this pattern is that both of these orders are obligate aquatic groups. So they spend a lot of their lives in water and then emerge as adults, mate, and return to water for the rest of their life cycle. So it's possible that they're somehow using frequency and sound pressure level to make some sort of habitat selection decision, or it influences their behavior in some way that is ecologically relevant. And it might make sense in some sort of desert landscape like this, where you have many miles of really dry, arid landscape, and then just a thin little riparian strip. And riffles and pools are different, and they have different soundscape profiles. So it might make sense for these things to actually pay attention to the soundscape. But this is not everything in the system, and a really important intermediate predator are orb-weaving spiders. And so I decided to look at that for a part of my PhD as well. And so there are two species that were really common orb weavers. There are a couple of others that aren't associated with streams, but these two species were always found associated with the streams. And they're from two different families. And so I measured the abundance of these spiders by creating these 4-meter transects, where I just waded in the stream with a flashlight in the middle of the night. These are nocturnal species, and looked for webs along the stream. And so this was some measure of abundance. But I also did a lot of behavior. So these are web dimensions. So if it's sort of an external phenotype, you can understand a lot about the hunger or the energy level and different things of spiders based on their webs. You can count the insects captured in the webs, which I also did, and then I also measured body condition. So it's usually a weight that is sort of calibrated to some standardized length. So you get some weight to length ratios, and that can be a metric of indirect metric of fields. And so I'm only going to focus on the abundance for this talk, but you can check out the paper, or I have a couple of other recorded talks if you send me an email that outlines all those other pieces, but we'll just talk about the abundance here. So I was pretty shocked to find that spiders increased dramatically, both species, in really loud, intense environments. And so axes are similar to before. The colors represent the two different species, and the points are raw data. And then the lines here, instead of having one individual line and then some sort of error bars around that, these are 95% of the innermost draws from the posterior in a Bayesian GLMM. And so you can just sort of see that here are a bunch of different draws, and so it's a different way to sort of get the same information. So you can kind of see the range of feasible predictions. But this is a pretty big increase over this range. And in fact, so this is a similar plot to the insect plot I showed earlier. It's flipped on its side, and instead of having a bunch of different insect orders, now we have all the different variables that were in the model. So at the top, there's sound pressure level. Below that, we have stream width, time after sunset, year, elevation, all these other things. And you can see from this that sound pressure level was the strongest predictor. So these are all these effect sizes are standardized, so they're directly comparable. And sound pressure level was the strongest predictor for both species, even stronger than temperature and wind speed and all these other things that are known to or are thought to shape orb weaving spider abundance and behavior. So that's pretty interesting. And there are a lot of different reasons we might suspect this to be the case. And so one is direct effect. And so the question is, well, can spiders hear sound? And that's sort of an open question for investigation. There have been some recent papers suggesting that other families of spiders can hear sounds further away. So spiders have a lot of tactile hairs that vibrate with close prey, so the wing beats of insects that are flying near them, and that's been known for a while. But these more long distance sounds are perceived differently. It's the difference between near field and far field hearing. But there is really recent evidence that orb weaving spider webs can actually transduce sound just as well as any of our best microphones. So going from an airborne acoustic stimulus to a vibrational stimulus, there isn't a lot of loss in that transfer from airborne to vibration. So it's possible that they're using their webs as giant ears and are actually assessing the value of the habitat or something like that with their webs or other organs. But at this point, that's all speculation. I don't have any evidence of that. Another way is indirectly, right? So noise can be shaping insects that are perceiving the sound in some ways, and that has some sort of bottom-up effect on spiders. And so we can look at that a bit. So I did measure the number of insects and identified insects in these spider webs, and we don't see a lot of change in those numbers with sound pressure level. But it is important, and more importantly, we don't see consistent changes across the species in a way that would just explain this pattern. But the caveat here is that spiders immediately consume prey that they're really interested in, and so most of the time when you're counting prey in webs, it's not really prey for consumption. So prey might be the wrong word there. And then the bottom right here is that same insect plot from before, and all the groups in the boxes are groups that I found in spider webs, and none of them deviate from 0, right? Meaning that if if insects are really changing their abundance, which is driving the change that we see in spider or spider abundance, we're not really seeing a lot of strong evidence for that here. So just to reiterate, both of these are ideas, but there's not really evidence for either of these with my data. But another way is top down, right? So I showed you before that both birds and bats decline as sound pressure level increases, right? And so if they're both of these groups and many of the species at our sites have been found to, and when I say species, the species of birds and bats have been found to have the exact species of spiders in their diets with genetic analysis. Right? So it's not evidence that it's occurring at our sites, but it is very possible. And it's important to note that there's all kinds of other scenarios here, right? The birds and the bats are also eating the other insects that the spiders are eating. So it's likely a lot more complicated than just one of the first three. And so, in the end of my PhD is where I just really started realizing how complicated a system is, and just describing, single species patterns with some environmental variables really wasn't going to tell you the whole story. And to try to put, all of these pieces together is going to be a lot more complicated and require some food web analyses and things like that. And so one of the projects we started working on to try to get at this is we caught about 200 of our 6 most common bird species, pictured here, and we got them in hand and put them in brown paper bags for about 10 minutes, and you can see the paper bags sort of ripped up here. They defecate in the bottom of these bags, and then you take your little ethanol vial and scrape up the poop, and then I took all this poop with me to Florida and extracted DNA from all of these samples with the goals of linking up this food web, and so that we can create something like this bipartite network where you can understand something about how commonly one species of bird might be eating different, you should say operational taxonomic unit rather than species of insect, because genetic analyses aren't perfect, but basically you can really link up these trophic levels. And a lot of the results I showed you before for insects were at the order level, but we've also blended up all of those samples that we ID'd to the order level and have extracted DNA from those so that we can understand some more species level patterns down the road. And so, in the end, the take home here really is that noise matters, and things change behavior, they change their abundance, they change their distribution just by changing the soundscape of a stream. And the future for me looking forward is really trying to integrate all of these things, because the world is really connected. And it's sort of a segue into what I'm doing now, which I'll just give you a brief, I just started, as Cinnamon mentioned, at the Hatfield Science Center here and working with NOAA and OSU on an ecosystem model. And so this is a network from Rizika et al. Of the Northern California current. And so the ties here, or the lines in between these boxes, represent some sort of strength of consumption of the higher trophic levels on the lower trophic levels. And so you can start to connect the world in a more complicated way than just single species models. And so the model I'm building has about 106 functional groups. Some of them are pictured here in these silhouettes. And it's called an end-to-end ecosystem model. And that is another, in fisheries, it's sometimes called a physics to fisheries. So upwelling physics drives phytoplankton production, which works its way through the food web all the way to fisheries. And so here's sort of the spatial domain of what we're interested in. There's the three bathymetric lines going down the coast are subdividing our world into spatial domains for the model. And then there are 5 latitudinal bins that you can see labeled here. And so we have these different spatial domains that we're filling in with abundance information or biomass, really abundance from a lot of different transects from NOAA and others. And so that's what I'm spending a lot of my time doing, is taking all of these data sets and either inputting the biomass from those data sets, or calculating biomass from those and incorporating that into this model. And another component is the diet matrix. So similar to that network I showed you before, you build a diet matrix where you enter the consumption for each individual functional group, how much they're consuming the other groups. And obviously this gets really complicated because diets can vary over space and time, and diet data those are really hard to get. And so there are a lot of things to work out with this model, and we're really just at the beginning stages of it. And so I'm happy to answer any questions about this too, but just note that there's a lot to do here, and it's not a lot. There's a lot more to do. But the goal eventually is to incorporate different scenarios. So you can, once you have a working ecosystem, and so by working I mean mass balanced, that matter is not created or destroyed, and so basically energy is moving through this ecosystem in a sustainable way. And then you can force different climate change scenarios or different management scenarios on your ecosystem to try to assess different things, such as what's going on with endangered stocks of Pacific salmon, or whatever else you might be interested in. So with that, the text here got a little messed up. There are a lot of people to thank for the PhD work that I did, and we had a huge field team, a lot of collaborators and things like that. And yeah, with that, I will take any questions.
All right. Thank you so much. I am having a little trouble with my camera, so you just got to hear my voice, so forgive me for that. I wanted to say thank you, Dylan, for working us through that, and doing that without your notes was very impressive, I must say. There was a lot of information that you shared with us, so I appreciate that. You're getting applause. You can't necessarily see it, but it is coming in on the chat and in some of the information there, so I just wanted to thank you. I get a couple of questions here that I wanted to ask, and I know that we're getting close on time, so if folks need to drop off, thank you so much for joining us. I appreciate it. But Dylan, I know that you also are speaking to our students here very soon, so I won't hold you for too long, but I'm going to ask you this question that just came in. Have you worked with natural sounds and night sky programs of the National Park Service? They've done a ton of recording.
Yeah, so let's see. Yeah, I guess none of them are listed here, but yeah, absolutely. So we've worked with them on both light pollution and noise pollution stuff. Kurt Fristrup was sort of leading that for a while, and he's retired now. But yeah, they're a good group.
Nice. And so with that, Dylan, I just wanted to say, I hope that you come back and share with us the work that you're working on now. I think there's lots of folks that'll be really interested in the outcome. Do you have access to chat in the format that you have? I'm wondering if you want to put your email up so that if people want to connect with you, they have some way to do that.
Yeah, I think I'll have to stop sharing because I couldn't see it. Okay.
So if anybody has questions, they can reach out to Dylan. He's going to put his information into the chat. There it is there. And so you can follow up with him. For everybody that's still with us, thank you so much for joining us. I hope that we see you next week at the Markham Symposium and supporting our grad students. They've worked really, really hard to get ready for this event, and we're excited to see what they're up to. Again, Dylan, thank you so much. You are again getting more thanks, and that was great. Thank you for sharing, coming in. So appreciate that as well. So everybody, I'll see you next week. Thank you so much. And Dylan, take a little break before you meet with the students, get some water, breathe a little bit, and hopefully we'll see you face-to-face at Hatfield sometime very soon. So thanks, everybody.
Thanks, Cinnamon. Thanks, everyone else.
Bye now.
Bye.